you.
Rauri and Sushma, I just wanted to check if you guys are okay if we put up the recordings on, online. We didn't actually take consent. We didn't put that in an email.
Yeah, it's fine for me.
Okay.
Yeah, it's fine for me as well.
Okay, cool.
I have to kick my dog out.
I know I'm in the same situation.
Hey, I keep meaning to add a nice backdrop. Haven't gotten around to it. I like yours, Rory. It looks good.
Thank you. I'm longing for the field.
Yeah, all of us.
And a quick note to the attendees that, please type your questions in the Q&A box.
Okay, we are going live.
Yeah.
Okay, good evening and welcome to the penultimate day of the Humboldt Day India events. These events are being jointly organized by Dr. Robin Vijayan from IISER Tirupati and Professor Ramakrishnan and I, Vivek from the National Center for Biological Sciences. These events are supported by the Museum and Field Stations Facility, NCBS, and the Biodiversity Collective. Today, we have Dr. Jayashree Ratnam, Associate Director of the Wildlife Biology and Conservation Program at NCBS, as the moderator. Dr. Jayashree is interested in the ecology, function, and the conservation of tropical savannas, forests, and grasslands. She is currently involved in research investigating the history, assembly, and the reclassification of Asian tropical dry biomes and long-term responses of tropical forests and savannas to ongoing and future climate change. Welcome, Jayashree.
Thank you, Vivek. And good evening, everybody. Good evening to everyone who's here with us this evening, and a very warm welcome to our speakers. I'm really pleased to be able to introduce our first evening, Professor Sushma Reddy. She's Associate Professor and Breckinridge Chair of Ornithology at the University of Minnesota. Sushma primarily investigates the evolutionary history of birds. Combining genetic, phenotypic, and geographic data, her lab addresses questions such as species limits and phylogenetic relationships, as well as synthetic analysis using phylogenies to infer phenotypic evolution, biogeography, and classification patterns. So I am really excited to hear what you have to say to us today, Sushma. And with that, I'll hand it over to you.
Thanks, Jayashree. I'm gonna share my screen now. Can everyone see this?
Yes.
Okay, great, thanks. Thanks everyone. Thanks to the organizers for inviting me. It's so lovely to see all these familiar faces, people that I have spent a lot of time with when I come to India, and this is, it's been more than a year this year for me to be there, and so that's the part of the pandemic that's really been affecting me the most, that I don't get to travel and get to see all of my friends, but I guess we should be grateful for Zoom for bringing us together in a different way. I'm gonna talk to you today about my project on the birds of Madagascar, and the organizers asked me to talk a little bit about what motivates my research and how I got to do the kind of, to research the kinds of topics that I do. And so I thought I would start a little bit with my, my own history and how I got to study the birds of Madagascar. Madagascar has always been this like exotic place in my mind. It's always this, I grew up fascinated and watching lots of documentaries about far off places, and Madagascar really illustrates this you know, isolated island on the side of Africa. I can't seem to get my cursor to point to anything. And it has a number of really exotic animals and plants. And so, sorry, I'm gonna see if I can make this smaller. If anyone can, if one of the organizers can make the little presenter box smaller, I can see my screen a little bit better. So the thing about Madagascar that really got me excited at an early age was it illustrates the concept of endemism better than anything else, right? So lemurs, for example, are only found in Madagascar and nowhere else in the world. And this was something that just kind of blew my mind at an early age and really brought me down this path of biogeography. Baobabs, all but one species of baobabs are only found in Madagascar, and look how bizarre they are as trees. And so this idea of kind of exotic and strange and only found in one place was really what drove me to study, to really dream about studying Madagascar. Can't seem to control it. Some of you younger people might associate Madagascar with a series of movies that I'll be honest, I was a little bit offended by at first because none of the main characters are actually Malagasy. And so this was something that I had to kind of, you know, push away from my mind until, as I was doing my research, I realized that maybe the writers of this movie were actually taking this early model that G.G. Simpson wrote and making it into a big Hollywood biogeography joke. G.G. Simpson, for the students out there, is one of the great evolutionary biologists who was one of the founding members of the modern synthesis that brought different kinds of ways of studying evolutionary biology together. And he made this key observation that you see quite a huge variety of mammals in Africa, right? Elephants, lions, zebras, lots of really big, charismatic megafauna. But just on a short ferry ride away is Madagascar, and there are not that many mammals there. So what happened? Why aren't there more mammals on Madagascar? And so he came up with this idea of a sweepstakes. This idea that only a lucky few winners were able to get to Madagascar. They were able to win a lottery ticket, take a cruise ship over to Madagascar, and be able to diversify there. So they were lucky enough to get to this new place that had lots of resources, and they were able to really diversify a lot. And so Simpson's also the first person that came up with the idea of adaptive radiation, a concept that was later taken over to illustrate kind of the most dramatic examples of evolution. And as someone who was interested in birds, I'm primarily interested in you know, what's going on in the birds in Madagascar. And so I realized as a student, how do we make this smaller? Can someone do that? I don't know if the audience can see my full slides because this panel is a little bit too big.
Yeah, we can see, Sushma. We can see the full slide.
Great. Okay, good, thanks. Then I'll just pretend I am too. Oh, shit. I can't go back. Sorry. Going forward. Give me 1 second. Let me just compose myself again. Okay. So Madagascar, in terms of its avifauna, was, you know, even just 10 years ago, still a bit of a mystery. So we knew that there were several distinct species of birds. For example, as illustrated in this textbook, the vangas that have these crazy bill shapes and forms. And we thought that they were another adaptive radiation, but we didn't actually know what their, much about who were vangas and what their phylogenetic relationships were. We didn't really know much, but they were kind of thought to be an adaptive radiation. And then the other remarkable thing about Madagascar is it doesn't actually fit some of the models that we should expect based on island biogeography. So it's a large island, and a large island, if we make a prediction that the size of an island, larger islands can support more species, we should expect to see more species, but instead what we find is Madagascar is kind of off the chart of what our expectations are. It has fewer species than expected. So this brings up a bunch of questions that were still unanswered even just 10 years ago. These are, there are some distinct forms, but how are they related to other things? Is it really less, is it more depauperate than what we should expect of a biodiversity hotspot? And as I said, it was still very much understudied. So my intro, my way of getting into Madagascar and fulfilling this childhood dream of going to these exotic places really came to be when I was a postdoc at the Field Museum. This happened to be a place that has the best modern collection of Malagasy birds, largely through the efforts of Steve Goodman, Tom Schulenberg, Mary-Jeanne, Rahel Lilalo, and other people who have been serving in Madagascar for the last 30 years. So they've been continuously going through and categorizing where things are and bringing in new samples so that we can study them in modern ways. And so when I was a postdoc, I was working on the Bird Tree of Life project, and I just happened to get this opportunity to study the birds of Madagascar, and I jumped on it. And I was part of a study with my collaborators to produce the first phylogeny of vangas. And this is really exciting, not just because this was a group that has fascinated me, but also lots of ornithologists for a long time. Here's a group which has quite a diversity of bill shapes and sizes. We thought they were an adaptive radiation, and this is the first study that was able to bring phylogenetics into it to show conclusively that they do share a common ancestor. So even though these birds are quite different looking, they all come from a single lineage that colonized Madagascar about 15 million years ago. And from as soon as they get there, they diversify into these different points. And so the power of phylogenetics is really illustrated here, where we can use this to show that this radiation is a radiation. There actually are more species in here in this group than we thought when we first started the study, which is also really interesting, which makes the disparity of their ecologies even greater. And we can use the phylogeny to look at how their adaptive traits evolved. So this was really an exciting way to start off working in this group, working in this area. But I wanted to also continue looking at the other lineages in Madagascar. And so I developed this program of looking at Madagascar in a comparative biogeography context. So let's look at the entire avifauna of a region and ask questions such as, how did these birds get here? And once they got here, what happened? How did they diversify? And in what ways did they diversify? And why are some groups more diverse than others, right? And so not every group that got to Madagascar is as dramatic as the vangas. What makes vangas so special? What makes some groups more special in terms of diversity? To give a little bit of background and to begin to address how birds evolved on Madagascar, we have to talk about what the origins of Madagascar itself as a region. Madagascar was once part of the supercontinent of Gondwana. So it's a continental fragment that broke off from the rest of Gondwana, was last connected to India 88 million years ago. and has remained an island ever since. So for much of the modern history of modern animals, and it has been isolated, has not been connected to anything else, and has been roughly in the same place close to Africa, about 400 kilometers away, for at least the last 80 million years. So this is, it has a long history of isolation, but it's also a large island that supports diverse habitats. And so we can make a lot of predictions about how animals got to Madagascar, how they dispersed over, and what might have happened once they got there. So given its proximity to Africa, we should expect that Madagascar is a small subset of the birds in the closest mainland. This is a prediction that we can make from island biogeography theory. And if we look at the mammals, again, contrasted with the example with the mammals, this is kind of what we see with the mammals. So all mammals on Madagascar are only represented by 5 lineages across the mammal tree of life. So, and of the non-flying mammals, this means that there are only 4 times that 4 lineages that got to Madagascar across the Mozambique Channel and successfully were able to colonize and diversify within. So even though today we see about 250 species of mammals on Madagascar, these were originally only 4 lineages that came over. So why are there so few? You know, we should expect, since it's not that far away, that there are more things coming from Africa. It turns out that maybe the way that the wind and the oceans were moving throughout much of the Cenozoic actually didn't favor dispersal from Africa to Madagascar. The winds were blowing in the wrong direction. They're actually blowing from east to west rather than west to east. And so that could be one explanation for why we don't see more things from Africa in Madagascar. So I said in mammals, there are only 5 lineages, sorry, from the mammal tree of life. On birds, the story is quite different. And we should expect a little bit of this because birds do fly and can get around a little bit more. This figure here is showing you the bird tree of life. You saw this figure yesterday from Walter Yetz's presentation. And what I've done here is just put arrows on the lineages that are found in Madagascar. And you can see that their Malagasy lineages are from across the bird tree of life. They're from all different parts. They aren't kind of segregated the way it is in mammals. But we can also kind of categorize them in 2 different ways. Species that got to Madagascar and just remained the same species, failed to diversify within, and species that got to Madagascar and subsequently were able to diversify using the resources in the island. And so if we look at that, we actually end up having two groups of roughly equal numbers of species. And this is a nice way to set up a model to test some key questions in evolutionary biology, right? So we can take these 2 groups and say, why are some of them more diverse? Why do, what might predict what drives diversification? Is there something inherent in the ones that are speciating in Madagascar? Because remember, the environment at this point is the same. They're all subjected to the same environment. So why do some lineages why are they able to harvest the resources in different manners? So the first question we looked at was to address maybe there's something inherent in the way that a certain group speciates that might drive diversity, right? So we calculated speciation rates for the source clade, the clade that the Malagasy lineages are nested within, and we plotted them against the number of species that are actually in Madagascar. And what we found was actually that species diversity on Madagascar is not correlated to the speciation rate of the source clade. You have a whole bunch of lineages, the ones that are highlighted here, that these are the lineages that failed to speciate, right? So they, and some of them have very high speciation rates globally. So across the world, they speciate pretty readily, but in Madagascar, they fail to speciate. And actually what we find is the species with the highest diversity on Madagascar tend to have lower speciation rates in their clade. So the most diverse group, the Vangas, I already talked to you about that. This is a group that has a fairly low speciation rate, but maybe something about how and when they got to Madagascar is leading to the high level of diversity we see there. The second most diverse group in Madagascar, the Bernierids, this is another phylogenetic mystery that was really interesting. So the birds that are in this group were actually birds that more than, that in all traditional classifications were placed into three different families, three different widespread families, the bulbuls, the babblers, and the warblers, because they kind of look like these members of these other families. And it turns out they're not part of those widespread families at all. They're actually part of an endemic radiation that are, and is the result of a single colonization into Madagascar that diversified into these different forms. And so again, another dramatic example of phylogenetics kind of solving the mystery of how these different forms came to be. And it illustrates again that, you know, sometimes there's a lot of convergence in how bird forms are in terms of how they're feeding, what they're feeding on, where they live, and that driving trait evolution in birds. And so it's important to use all of this in a phylogenetic context for us to be able to study how they've really diversified. So getting back to our biogeographic question of how did birds get to Madagascar, I said before, the prevailing theory based on island biogeography is that the closest, most of the birds on an island should come from the closest mainland. Well, in Madagascar, we find that actually it's about evenly split between Africa, which is the mainland, the closest mainland, Asia, which is parts of Asia which are much more distant, but on the other side, the other source areas that are possible, and then about a third, which we're still unsure about, and this could be because they're either really old relictual lineages in Madagascar, or we're still working out the phylogenies for this. And so even though Asia is much further away, and the Indian Ocean has the vast amounts of space with no land masses, it turns out that there's a fair number of representatives that have moved around across these ocean islands and got to Madagascar. To illustrate this with some bird lineages, we, these are some of the endemic birds in Madagascar, and I'm just gonna kind of highlight some of the cool ones that show clear affinities to one area or the other. So this yellowish bird with a wattle is Cetes. They're actually really closely related to broadbills and closer to Asian broadbills than they are to African broadbills. And so we think they have African, Asian origins. Next to them are the kuas, which are a lineage of ground cuckoos. And we think that they're closely related to ground cuckoos of Asia and quite different from the cuckoos in Africa. Things that we are fairly certain have an African origin are things like the ground roller, this guy here with the bluish head, kingfishers and weavers, the foodies that are endemic to Africa and some of the, to Madagascar and some of the surrounding Western Indian Ocean islands, we think have an origin from the weavers in Africa. And then there's some really strange birds still that we're working out, such as the mezites, this brown bird here, this big brown bird on the left side. These are birds that have long baffled ornithologists, and it took the Tree of Life project basically to really uncover their closest relatives are. And it turns out they're closely related to sand grouse. Again, another long very distinct lineage on the bird tree of life, and one that we're still kind of figuring out how they got to be. Are they a relictual population on Madagascar, or did they actually originate through some ancient divergence event in Gondwana? So now I want to focus on whether we can take this issue of how they got here, how they got to Madagascar, and see what that predicts about their diversification, right? And so I mentioned that wind currents and ocean circulation currents were really important in kind of explaining why mammal diversity was so limited in, or the number of lineages on Madagascar were so limited. Does this explain something in birds, right? And so for the early part of the Cenozoic, we have the wind conditions blowing from Africa into Madagascar, and then halfway in the Miocene, you have wind conditions blowing in the opposite direction from Asia into Madagascar. And so is that what we find when we look at the origins of Malagasy lineages? And actually what we see is that's not entirely the case, right? We have fewer African lineages when wind conditions are favorable coming from Africa. And we actually have more African lineages when wind conditions are going the opposite way. So that's a hard one to explain. And perhaps that could relate to something that Rosie Gillespie was mentioning with her critters in Hawaii, that maybe some of these are actually the result of catastrophic events and not just kind of what the normal patterns are. And then finally, is there an aspect of when and how they got to Madagascar that kind of really leads to whether they radiated or not? We should predict that if they got to Madagascar earlier, they should have more time to diversify, and that's exactly what we see. More lineages that got to Madagascar earlier are the ones that that diversify. But what's interesting is that it's about half and half, right? Only a little more than half of the species that got to Madagascar early on have gone on to diversify, and about a little less than half have failed to diversify. So what's going on there? It's possible that there's a high rate of extinction in some of these lineages that we haven't factored into our models, but it's an intriguing question that we'd like to look at more. Now I want to focus a little bit more on the last question that we are, one of the questions we're focusing on in my lab, which is to contrast how adaptive radiations really diversify. So in this context, I'm going to focus in on the two largest avian radiations in Madagascar, the Vangas and the Bernierids. And these are the two most species rich. Vangas have twice as many species as the Bernierids, but they share a lot of other similarities in terms of when they arrived in Madagascar. And so we can use these two to contrast how groups do different things, even in the same environmental setting. So we see that the timing of colonization in Vangas and Bernierids is roughly similar. And in Vangas, what we find when we reconstruct the biogeographic history is that there's a higher likelihood that the Vangas colonized via, the Malagasy Vangas colonized via Africa, but at the same time that they were colonizing Madagascar, they were also colonizing Asia. So the closest relatives of the Vangas in Madagascar are in Africa, but also colonizing into Asia at the same time. And so it's kind of something that's happening simultaneously. And then in the Bernierids, the situation is a lot more complicated, where the closest relatives of the Bernierids is actually this really bizarre species, Donychobius, that only lives in South America. And so we think that the best explanation of this is that the ancestor of Bernierids were actually much more widespread. And so there was a lineage that was much more widespread that colonized Madagascar and eventually evolved into the Bernierids and also simultaneously got to South America and evolved into Donoclobius, and their closest relatives together, their closest relatives are the locustelids, which are grassland birds found from across the old world. And so this is kind of a neat illustration, again, with the Bernierids. What's really interesting when we think about where they live, they not only they're also different in terms of their habitats from their closest relatives. So the locustelids are grassland, bush living species, and the Bernierids on Madagascar are almost exclusively forest dwelling. So there's, again, the shift in their behavior once they get to Madagascar. Some of the predictions of adaptive radiations is that in the model of ecological opportunity, species get to a new area, they begin to exploit niches, begin to exploit resources and start filling in niches. And as they start filling in niches, the speciation rate should actually decrease. And so a model of adaptive radiation through ecological speciation, ecological opportunity predicts that speciation rates will decrease over time. And that's exactly what we see with both the Bernierids and the Vangas. And so this is kind of a nice, result that fits the models that we should expect. Now I wanted to look at also how this question of why some groups are more diverse and how they evolve. And I keep talking about this idea of adaptive radiations, and here's a nice illustration of why this is an important concept in terms of kind of illustrating the dramatic ways that evolution works, right? So Darwin's finches, which you guys have all heard of, Hawaiian honey creepers, and now taking their place also the Malagasy Vangas, where they, you see changes in morphology, changes in bills, especially because of what they're eating and how they're eating. And so we can make inferences about how evolution in these features has happened in response to new resources. And here's a really great study that illustrates what the possibilities are. This is a study that looked at the cranial morphology of Hawaiian honey creepers and Darwin's finches and their closest relatives. And what they're looking at is the amount of variation or disparity across these groups. And the way to read this is a plot of principal components, the amount of variation across these groups, and the blue polygon, the solid polygon there represents the variation in the Hawaiian honey creepers. And if you contrast that to what their closest relatives, their outgroups are doing, the green polygon there where it says non-Hawaiian honey creepers, you see that they're quite different. The Hawaiian honey creepers have shifted in their morphology and have kind of taken over a part of morphospace that's quite different from their ancestors. And the same for Darwin's finches. And so this sets up an expectation or prediction we can make about adaptive radiations, that they're doing something a little bit differently than what their ancestors did, differently than what their closest relatives are doing. And for us to test whether this is also true in the Malagasy species, we set out to use museum specimens to measure different features. I had the help of lots of students who came with me on these trips. And we looked at different features of bills and feet and color. And what we found was very much exactly what we see in the Hawaiian honey creepers and Darwin's finches, that there is this shift in bill shape and size associated with the colonization of Madagascar. And so there is something different happening in this lineage, in this area, than they were able to do in other parts. I had a student who took this the next step and actually used 3D photogrammetry, which is a very simple way of taking digital photographs and stitching them together to make 3D models. And with these models, we can actually look at 3D shape and disparity. And again, he shows very similar results and even more dramatic results of disparity in terms of the shape of these bills being very different in the Malagasy groups versus their close relatives in other areas. And this is something that clearly also seems to have an association with behavior. So the rate of change in different lineages seems to be associated with the behaviors that they are using these bills for. So birds that are using their bills for probing are evolving at a faster rate than birds that are doing what the ancestral behavior was, which is just gleaning and picking things off of the substrate. And so behavior has a strong correlation to driving these adaptive changes. And so this kind of helps to illustrate again that some things, ecology drives evolutionary differences in these birds. We see a lot of a similar trend in the Bernierids. The Bernierids are something we don't understand this group enough. We don't have a good sense of what all their ecologies are, so we don't have good explanations for why some birds have really much longer bills than others. But we're starting to get there. We're starting to examine this more closely. And now that we know that they're an endemic group, we can make these comparisons across these different species in a way that's more meaningful, and to look at differences across species within this radiation. But again, if you compare them to their outgroups, they seem to show the same kind of shift in their morphologies from their relatives on different areas. Another thing we're doing in my lab is spending a lot of time looking at the possibility of hidden diversity. So I mentioned earlier that the number of species in Madagascar is less than we would predict given its area size. And perhaps this is because we actually haven't recognized all the species. And just within the two groups of Angas and Bernierids, we have using detailed analyses of genetics, ecology, geography, and comparing morphological features, et cetera, using an integrative taxonomy framework, we've been able to increase the diversity of Madagascar by more than 8 species. So this is pretty dramatic change in our understanding and very important for issues of conservation. So we have lots of plans. We have a whole bunch of data for other lineages, and we're starting to process them to look at them in the same kinds of ways that we've looked at the Bernierids and the Vangas. But one of my major future goals is to really help address this issue of decolonizing science and supporting Malagasy scientists in the ways that I can bring in, or Western scientists can bring in. I was able to study these groups because I had access to museum collections in the United States and in Europe. I was able to travel to these places to make use of their collections. And I am able to travel to Madagascar to get new collections. But that's not true of scientists in Madagascar. And so one thing I can do in my lab, or we can help support, is making the data in these collections accessible, and give kind of repatriate data back to these countries, because conservation is a global issue. It is a global heritage that we need to be putting efforts in different ways. And so this is something that I feel strongly about, and I feel like much of my research has been using museum collections, and I think the importance of museum collections can be really be illustrated in multiple ways, and here's a great way of that. We do lots of different things. Like I said, we're looking at the diversity of birds in Madagascar in multiple ways, and if students have questions about this, I'd be happy to talk more about some of these issues later on in the question and answer. And just want to end my talk with a thank you. I've been really fortunate to work with a series of really great people who have helped me bring these projects together. But I want to dedicate this particular project to a former student of mine, Lenika Stroger, who we lost to COVID recently. She was just, a great illustration of a hardworking and incredibly amazing. There's no other word for it. She was an amazing scientist, and it's really just unfortunate that this ruthless virus can take away some of these real treasures in our lives. So she was one of my students. She was one of my first students, and she helped me really like bring some of my ideas of Madagascar together. And so I think she deserves extra credit for that too, in addition to all the other wonderful things she did. So I want to end with a dedication to that, to Monica. And I'm going to stop before I get more emotional. So thank you for your attention. And if you have any questions, I'd be happy to take them.
Thank you, Sushma. That was a wonderful talk. And, you know, thank you for telling us about Leinica, and I hope her soul rests in peace. At this moment, we've actually kind of run over time, Sushma. So I think no worries, but we do, that's why we've set aside 20 minutes at the end of both the speech. We hope to take questions for you then. There are some very nice questions that I'm seeing in the question box, but we'll come back to you with that after Rory's talk, okay? With that, I'd like to introduce our second speaker for the evening. And yeah, and this is Prof. Welcome today to Prof. Rauri Bowie. He's Prof at the University of California at Berkeley. Rauri and his lab study evolutionary biology, primarily of vertebrates, with an emphasis on species endemic to tropical biomes and Western North America. So they combine field, museum, and genomic approaches to document and delineate biological diversity and to study the basis of animal diversity, evolution, and biogeography. In the course of their research, they apply phylogenetic, population genetic, and genomic approaches, working with data sets overlaid with analyses of acoustic and trait variation and species distribution models to examine spatial and temporal patterns of diversification. His research is conceptually as well as geographically broad and addresses multiple fundamental questions in evolutionary biology. And once again, I'm really excited to hear what you have to say today. And with that, I'll hand it over to you.
I don't know what, somehow can everybody see my screen?
Yes.
Yes.
Yeah.
Something happened with the first slide, sorry. Okay, I'll try go as quickly, but you can read the screen, hopefully. So today I'm going to talk about sky islands, and I'm going to try and cover these 4 topics. I just want to make sure everyone can see my cursor and the individual topics.
Yes, we can see.
Looking good, looking good.
Okay.
Yep.
Okay, it's looking a little funny on my computer. Okay, so I'm going to introduce you to the Sky Island system, and then I want a little bit talk about a model that we've developed that may explain some of this diversification. And if I have time, I'm going to touch on the use of whole genomes in this kind of context and a little bit about trait diversification. So I want to start off with this slide because I think it's important to get a perspective of just how large Africa actually is. On many maps, it's sort of compressed in the middle. And just to give you an idea, this is India over here, the United States over here, and China over here. So it's truly an enormous continent. And it has this really interesting variation in vegetation with this very large desert, the Sahara in the north, these tropical rainforests, lowland tropical rainforests. And of course, in context of today's talk, we're interested in the Sky Islands themselves. So just as Humboldt was one of the first people to climb some of the South American Sky Islands and notice the stratification of vegetation and some of the beautiful work that Robin's done on the Western Ghats in India, which most closely resembles the African Sky Island system. In Africa, we see this dramatic stratification as well. So here's Mount Kilimanjaro with the ice cap. Today, it's nearly completely disappeared as climate has changed. This is at 4,500 meters on Africa's second largest mountain, in Mount Kenya, where you see these giant lobelias and this kind of tussock grass habitat. And then we get this interesting phenomenon where in the tropics, we get rainforest sort of between 1,000 and 2,000 meters. And then in Southern Africa, as latitude compensates for altitude, tropical rainforest actually comes all the way down to the sea. It reaches the coast over here. One of the interesting features about the African montane system is that it's naturally a sky island system, and it's divided into these 7 different areas of endemism. So this is a distributional overlay of the nearly 300 birds in Africa that breed above 1,500 meters. And these are the areas of endemism. We have about a 5,000 kilometer long chain that goes from Ethiopia through Kenya, the Eastern Arc Mountains of Tanzania, the Albertine Rift, and then into this chain that connects Malawi and Southern Africa. And then we see 2 isolated areas, the Angolan highlands and the Cameroon highlands over here. One of the interesting things about mountains, and this is true globally, but especially true in Africa, is that if we just take a phylogeny, in this case, it's the African green bulls or bulbuls, there's 65 species, and we divide the branch lengths in the phylogeny into quarters, one of the things that we notice is that the youngest lineages, the youngest quarter of lineages, tend to cluster in these mountainous habitats. So we see a correlation between where the young lineages occur and mountains. And one of the things we've been doing is through our fieldwork, we've continued to describe new species that occur in these kinds of habitats. Here's just 3 examples: a new batter species, a new warbler species, and a paper we currently have in review that's describing 2 new cisticola warbler species. So what I'm going to do for today is I'm going to just focus in on this particular area here where I've done the most work, the Eastern Arc Mountains. I'd love to tell you more about how all of these different areas connect and their own special features. But the Eastern Arc is an area that's very intriguing. It's a series of 13 different sky islands. They're at least 6 million years old, so they're very ancient, and they have a fascinating biogeography. One of the things that puzzled me when I was still a student and starting to think about questions to pursue for my PhD was this figure generated by Jon Fieltze in Denmark. And this example here shows you species richness with the redder the color, the more, or the greater the number of species. And this shows you endemism for the same bounded box. And what you'll notice is these 3 mountains, including potentially this one in Malawi here, have very many more species than what you would expect by chance. So they have a greater, and especially endemics, they have a much greater number of endemics than what you would expect for their area. And so I was fascinated, why did that happen? What could be some of the explanations that underpin that pattern? So the first thing we can try and do is divide up the distribution of species by looking at what I call neo-endemics, so species that are younger than 3 million years, and paleo-endemics, sort of ancient species, or if you think about it in the context of a phylogeny, species that have very long branch lengths across the phylogeny. And we can see that these particular mountains, this Udzungwa, the Uluguru, and the Usambara, have both a high number of these very old species, and the whole of the Eastern Arc system has a high number of relatively young species. And so it's probably this combination that's playing an important role for generating this pattern. So let me introduce you to the Eastern Arc Mountains a little bit more. This is the chain of mountains that runs from just in Kenya over here down towards Malawi. This red line over here represents this digital elevation profile that's depicted here, going from the Usambara Mountains here in the north down to these Udzungwa Mountains or highlands over here. And you can see these are the individual sky islands. If we just draw sort of the digital elevation model, we extend it to this isolated highland over here, you can see again, this is this transect over here, a deep valley, similarly a deep valley and a deep valley, suggesting this Uluguru Mountains quite isolated. And so these are not human-induced fragments. This is a consequence of the climate of East Africa. And so these areas in between here are extremely dry, shrubland, very, very different to montane forest. And so we see these natural isolations of these different highlands. And this is the largest single gap. This is about a 100-kilometer gap separating out the Usambara Mountains here from these Nguru Mountains. If we focus in just on one of the groups, one of my favorite groups of birds that I've worked on, the Eastern Double-Collared Sunbird Complex. When we started work on this complex, only 2 species were recognized, one in the north here and one in the south over here. And as we were able to accumulate more samples and do our research, we now recognize 5 different species, which are color-coded over here. And possibly this one may represent 2. And that's really important from a conservation perspective, because you can imagine that your goals are very different if you have 1 species distributed over 5 mountains versus 5 species each restricted to their own particular mountain. And then we see interesting differences in song variation with respect to some of these groups. But again, the largest divide separating out the northern group from the southern group over here is across that large 100-kilometer gap between these 2 mountain systems here. So perhaps as we would expect when we look at the digital elevation profile. We can extend this work to many different other bird species. This just shows you an example of 6 of them with the distributions of the different clades depicted here. And in most of these species, this large barrier, this 100-kilometer barrier is apparent to be the most significant biogeographic break. And we can summarize this further. This just shows you the 15 groups of birds that we've worked on to date. And for 13 of those 15 groups, this gap over here represents a geographic barrier. For 8 of those 15 groups, this mountain comes out as isolated. And then these other 2 barriers, which have been suggested in the literature to be important for things like plants and frogs, turn out to not be as important with birds, with only 5 or 3 species showing biogeographic breaks here. But one of the other interesting things, although this is based on genomic data, just highlighting here, if we look just at this barrier over here, one of the other things that sequence data allows us to do is gain an estimate of relative divergence, which could act as a surrogate for time of divergence. So if we look at this figure here, we can see that in this green bull or bulbul clade over here, there's 12% divergence, and there's the full spectrum of divergence across this barrier for these different groups of birds, all the way down to Fullerborn's Alethi, which still has reciprocally monophyletic groups. So clades that are restricted to either north or south of this, but only very, very recent divergence. So we can ask, well, what having that pattern? It's unlikely to be purely a consequence of difference in population size. And so it's likely to be some other phenomenon. And given how old these mountains are, remember I said there was at least 6 million years old, it's likely that forests have expanded and contracted multiple times, and that may be driving some of this pattern. So hopefully this next figure works, but what I want to show you is just a movie going back from the present to 120,000 years in the past through 64 different climate slices over this time that we've reconstructed. So on this scale over here is going to be the present moving back in time. And what I'm modeling is the distribution of rainforest across Africa in that period of time. So here we go, going backwards in time. You can watch how these different areas, remember this is the Eastern Arc, this is Kenya, the Albertine Rift in Ethiopia up here, connecting to Angola and the Cameroon highlands over here. As these areas, and the darker the color, the higher the probability of rainforest being present. And you can see this contraction and expansion of these forests through the last 120,000 years. Oh, now we're going back up. Okay, we can summarize this information by calculating a statistic that's I call stability. And you can really think about it as just the habitat persistence through time. So if we divide each one of those slices into pixels and we ask how often was forest present in that pixel over each one of those 64 climate slices, we can calculate this metric, where a metric of 1 would mean that forest has been present in all 64 of those slices. And this just shows you the pattern over here. Here's the Eastern Arc system coming down here. Here's Lake Malawi, the Ethiopian highlands, the Albertine highlands, the Cameroon highlands, and Angola over here. And this is just blowing up the Eastern Arc section over here. So just this blowing up. And you can see that some of these highlands have retained forest for all of that last 120,000 years. We can also do things like building dispersal of rainforest trees by saying, either they can disperse something like 10 kilometers per 1,000 years or 20 kilometers. And what this essentially does is just reinforce this pattern, because it makes it less likely that forest is going to go extinct in any one of our pixels that we're modeling. So again, coming back to this question, it could well be that we see these pulses of rainforest expansion and contraction that's driving this. But now we want to know if that is the case, well, how many times have, for example, these mountains and these mountains been connected over this barrier? It seems unlikely given the deep genetic divergence that birds are just flying, so we can get an idea of how often they were connected and how might that explain this variation in sequence divergence. So we can make use of these hierarchical Bayesian statistical models to try and model this. And when we do that, this is just showing you for the mitochondrial DNA, we can quite convincingly show that there must have been at least 3 periods of connectivity between those 2 different mountain systems across this large barrier. One that explains these deeper levels divergence, one that accounts for these sort of mid-levels of divergence, and one that accounts for the more recent. And the differences, why there's this variation, is likely driven by differences in population size for these different species as a consequence of their ecologies, whether they're understory feeders or they're feeding in the canopy. And as a consequence of difference in population sizes, you get fixation of different alleles at faster or slower rates. So what I think is happening to drive diversity in this system is we're seeing these repeated episodes of an ancient expansion and contraction, division between Sky Island A and B, then they contract again, then they expand again, contract again, and expand again. And I sort of think of this as a pulse speciation model where both dispersal and then vicariance, the subsequent isolation, have played an important roles in shaping montane bird communities in the context of these pulsed, or if you like, cyclic expansion and contractions of these different rainforests. So this kind of model of expanding and contracting does a really nice job of explaining why we might see young species distributed through the whole system, but it still doesn't fully account for why do we see the ancient species only occurring on some of these mountains, but not all of the mountains. So what is it about some of these particular mountains? Well, one way to get at that question is we can use our same modeling framework. In this case, just modeling a cool and dry climate, a warm and wet climate, the current climate, and then summing across these 3 climate slices to see where forest is persisted. These are the mountains in the north, these are the mountains in the center, and the mountains in the south. And what this just shows is a higher probability of forest and a low probability of forest. And here we can be a little bit more specific when we can say that forest has stayed stable, or forest has become reduced, or even that forest has disappeared. And then we can make use of population genetics data using thousands of genes sampled from across the avian genome on both the macrochromosomes and the microchromosomes. And we can look at, for example, the distribution of several summary statistics. So here's just an example for 2 species. We can look at things like heterozygosity, things like nucleotide diversity, things like theta, and ask how this varies. So if we just look at autosomal loci, we can see that forests that are stable tend to have much higher levels of diversity than forests that are unstable, so where forest has disappeared over time. Similarly, we can look at other kinds of markers. These are sex-linked markers where we see a similar kind of pattern. And so what I think is happening in this system is that on one side, you have these pulses generating diversity as mountains connect, birds move between them, then they isolate, they speciate in allopatry. And then on the other side, certain mountains are much more likely to retain forest as a consequence of rain coming in off the Indian Ocean and hitting these mountains. And because forest has persisted for a long time, that's consistent with it not, with species being able to persist there for a long time. So you can think of it as an extinction filter. Forest gets lost on these interior mountains, and as a consequence, you keep losing species at a higher rate than what you do in these particular mountains. And so those 2 things together may well explain this level of diversity. So I just want to briefly talk about 2 different directions that we're busy going in now to try and exploit some of the data revolution that I kind of, as I think about it, and some of the new methods that are available. So again, one thing that we can do is make use of some of these new demographic models, in this case, this pairwise sequentially Markovian coalescent model, that for the first time really allows us to reconstruct the demography of species or populations over very long periods of time. So this is a mark at 10,000, Here's the last glacial maximum. And this is 1 million years ago. So we can really go back quite far in time. This is for a robin chat species endemic to the system. And these curves are different populations that represent these different mountains that I've indicated here in little black boxes. And you can see that there's a fair amount of difference. So some species have declined consistently. Some show a hump here in the last interglacial. And then one population shows a large expansion in population size in the last glacial. And we can break this down further by actually looking at individual models. And so what you can see is some of these very stable mountain systems that I've talked about. So these three over here show expansions and contractions in their population sizes, a little bit, but fairly consistent population sizes. Whereas some of these southern mountains, so for example, this one, and both of these over here show generally a long-term decline. And we think that the difference in these 4 southern mountains versus these interior mountains here is a consequence of rain shadow effects. And we know that from cores drilled here in Lake Malawi that between about 110 and 75,000 years ago, there was a massive mega drought in Africa. And so as a consequence, forest almost completely disappeared from much of this part of the distribution range for this species. And that could account for some of these trajectories. At least it's consistent with the core data that came out of the lake. And then I'm going to end off with just briefly touching on trait diversification. And I want to come back to one of my favorite groups of birds, this eastern double-collared sunbird complex. So sunbirds are, of course, Ossean songbirds. In other words, they learn their song. We can think of learned song as a cultural trait. And one of the things I want you to notice is the fair amount of variation in the song of these birds depicted over here. So birds in the north have these shorter songs. Some of them have slightly longer songs. And then this bird in the south here, Fullerborn's double-collared sunbird, has this really complicated song. But this is the figure that really struck me and what I've been trying to puzzle over. Your students, McIntyre, has played a significant role in helping understand. So here we're comparing 2 species, Moreau's sunbird over here with a short call and Fullerborn's sunbird over here with a long call. And when you compare between species, you'll notice that they're very different. Both, I'm just going to move this here on my screen, both in genetic distance. So they're about 6% divergent in mitochondrial DNA, and they're very divergent in their songs. But what's really fascinating is when you look within species. So remember, these are Sky Island populations, right? So we have discrete replicates for each of these species. And when you do that, if you look at Fullerborn's sunbird, you'll notice that as you increase in genetic diversity, song distance doesn't increase. Similarly, if you look at Moreau's sunbird, as you go to 2% sequence divergence between some of these populations, song divergence doesn't increase. So this is very counterintuitive than what you would expect for a cultural trait. Just think of human dialects. As we go expand in geography, we expect cultural traits to change. Yet when you look at this data, even at 4% sequence divergence, we see no divergence in song distance within populations for this species. And so why might this be occurring? Well, the first question, or the first thing we tend to think about is, well, maybe it's something different in terms of geography. Maybe it's some kind of ecological explanation and they occupying different habitats. And again, if we do reconstruct ancestral niches, or we project Moreau's sunbird onto Fullerborn's sunbird over here, or the reverse, we can see that their ecologies are extremely similar, and actually each one predicts the distribution range of the other. And so it's not something ecologically being driven. Similarly, we could look at bill morphology or beak morphology. And again, they have very similar beak morphology, so it's not some kind of morphological difference. So what else might be driving this? And so this is a figure that I like that sort of summarizes some of the different options. And it's got a great title, The Million-Year Wait for a Macroevolutionary Burst. And so these are the kinds of things that can happen. Here's Brownian motion, or is just cultural drift. You expect 2 populations that are isolated to change slowly over time. The opposite of that is you can have a bounded evolution, or what I kind of think of as stasis, where there's no change over time. They're remaining stable for a very long period of time. or you could see a scenario where you at some point get a rapid change in a trait over time, but it only occurs once and then you've shifted into a new regime. So my student Jay McEntee and a statistician were able to develop a model that allowed us to test among those different curves. And so here I'm just showing you the results for one song variable. This is the log of song duration. So it's an important variable because here you can see a short song versus a long song. So this is fuller-born sunbird, which has these long songs. Here's some others with a couple of long songs, and these birds over here have short songs. And what we were able to show is that using this model, there's strong support for this punctuated evolution model, or if you like, one pulse, in 4 of 14 different song traits we measured, and there's moderate support in a further 3 traits. And the places they occur in the phylogeny are over here, where we get a rapid switch to a long song, and right over here at this node, where we get a switch into these long songs from short songs over here. And so what's also interesting is that the mean of this is only about 1 pulse per one of these traits across the phylogeny. And so this indicates that the evolutionary mode of multiple aspects of these territorial song includes large jumps in these mean trait values, and then extended periods of hundreds and hundreds, maybe even millions of years of highly bounded evolution or stasis in the same aspects. And so we're seeing really quite interesting evidence of these sort of pulses of diversification in these traits in very long periods of stasis. And this explains why when we look at some of these isolated populations in these ancient mountains, which are different by such high degrees of sequence divergence that in many cases we would call them different bird genera, show no morphological variation that we can detect, and even in some cases, no variation in song. And so it's very hard to know whether to call them cryptic species or not when you cannot find this trait variation. So coming back to this example, we could ask, well, what might be driving some of this variation? And one of the things that it could be doing, this is some work that we published a few years ago. Here's Moreau's sunbird, the one with the short song over here. Here's fuller-born sunbird with the long song. They come into a contact zone right in these mountains over here. And when you model the climb for this zone, it's extremely short, this contact zone. It's only 6 kilometers in length. And so it's possible that this contact zone, this meeting of these taxa may have facilitated driving the divergence in this trait. There are several other potential explanations as well. So this just shows you gray is the using DNA to look at the hybrid index, orange is the change in song across this index, and then blue is the change in beak length, which is a little bit more diffuse. So I'm going to stop there. I think there's a lot of interesting things to still be done with traits. I'd like to thank many people and many museums who facilitated this work, but many people who've accompanied me in the field, as well as the National Science Foundation who funded this work. And I'm happy to take any questions. Thank you. Stop sharing.
Sorry. Hi, can you hear me? Yeah.
Okay.
Okay. Hi. Thank you, Rory.
Okay.
That was an amazing talk and a whirlwind tour of the Eastern Arc Mountains and diversification there. Yeah.
I apologize if it was a little fast.
No, it was fabulous. And we've sort of got a whole bunch of questions that are slowly starting to come in at the Q&A box here. Robin, shall I hand it over to you now to moderate this session, or Vivek, one of you? But should we do Sushma first and then Rauri, or do we mix it up? How do we go? I see a whole bunch of them coming in. Yep.
Yeah, I think some of the questions for Rauri are still coming in, and,
yeah, so
maybe we can start with Sushma and then.
Yeah.
Sushma, do you have? Can you see the questions in the Q&A box?
Yeah.
Should I read them out to you, or?
It's either way, it's fine.
Yeah, so some of the questions are upvoted, so if you could start from the top, that would be good.
Oh, okay, great. So Harshit asks, since India and Madagascar were pretty close in the past and then broke away, how much of its flora and fauna is related to India, like common ancestry, etc.? That's a great question, and that's something that we've as biologists and biogeographers have been debating for a long time. And there isn't an easy explanation. We're still kind of figuring it out. You know, one of the classic cases was using cichlids, because freshwater fishes are by necessarily kind of restricted to the landmass that they're in. And so there was a feeling that they would kind of preserve some of the connections of Gondwana, but more recent studies are showing that actually it's a much better explanation, because they're the cichlids in Asia and India are much younger than we thought, much younger than Gondwanan times, and so perhaps that's not a good illustration. There's some frogs that seem to follow the pattern of Gondwanan breakup pretty well, but again, these are things that are slowly coming in, and because of all the changes that happened in India once it collided with Asia. We think that probably a lot of the endemic biodiversity on the island when India was an island, probably got erased. And so maybe we have to look kind of underground for things that survived through all those things. Should I take the next question, or do we want one for Rauri?
Robin, it might be easier.
Sushma.
Okay.
Yeah.
Go ahead.
Go ahead, Sushma, for the next one. Oh,
okay, sure. So the second question is from Sahas, and he says, is there, oops, I lost it. Is there any,
third place, yeah.
Okay. Is there any fossil or subfossil evidence for multiple extinctions in a group that may not have many extant species in Madagascar? And that's also a really good question. So most of the fossil, or for birds, most of the fossil evidence is more recent subfossil material that they've been uncovering in caves, and there's a couple of recently extinct species, but not many examples of things that are like lineages that are no longer present. They're mostly there's like an extinct kua, extinct species of mesoites and stuff, but not anything that's like completely different from what we see there today. But fossils are another great area that needs a lot more work in Madagascar. Ashwin says,
I can just read the next question.
Yeah, shall I?
Sure.
I will just read it out, Sushma.
Yeah.
So Ashwin asks, could the species lineages that didn't speciate on Madagascar, despite having higher speciation rate, be the late arrivals and were posed by competition, less habitat area available? And was that because of?
Yeah. So that is what we find for some of them, but there's a fair number of them that came early on and didn't speciate. And so we're kind of examining the question of, did they fail to speciate because, or did they speciate and then they also have a high extinction rate? And so what we see, the end result we see now is low numbers of species. And so that's kind of the contrasting question that we need to examine.
There's another interesting question from Ashwin. Does the colonization and diversification of the Bernieridae, I hope I said that right, 11.5 million years, coincide with grassland expansion? And could that be the reason for clade relationships with South American lineages?
Yeah, that's a really good question. And I think that might be a situation. We haven't explored it in that angle, but what's interesting is also the question of grasslands in Madagascar. For a long time, we thought that there were no natural grasslands in Madagascar before humans, and that humans actually brought grasses to Madagascar. And I think that story might be changing as we get more information from pollen cores and kind of these relictual habitats. And so today, the central plateau of Madagascar is almost entirely human-driven rice paddies. And so it's hard to tell what that landscape looked like before humans got there. But it's likely that there was some grasses present, but most of the island was forested. And so what was available for Berniards to diversify into was a lot more forest land than grasslands. But what was happening in their close relatives in other parts of the world might have been expansion into grasslands. So yeah, that's really neat.
Okay, so this is actually a question kind of about approaches, and I guess it's addressed to both of you. So it's a general question. I wanted your views. This is from Harshit on using a combination of fossil data and present specimens. So I think he's speaking of ancient DNA, I guess, as I'm interested in this in the future. What's the best thing that can be done? Difficult question. Maybe Sushma, you can attempt, and then you can say something, and then Rauri. Thanks.
Sure. One of the things that we want to do, both for living species and fossils, is one of the things we can do is look at morphology, right? So that's a kind of data that we can get from both of them and compare them pretty easily. We're not going to, unless they're recent fossils, we're not going to be able to get genetic data out of it, but we can tell a lot about what an organism was doing based on its morphology. We can tell a lot about its evolutionary relationships. We can tell something about the environment it lived in based on where it was found and what the conditions were like in the past. And then we can use modeling, like Rauri showed, to kind of infer what the past looked like to be able to connect fossil and extant species together. Rauri, you want to add more to that?
Yeah, I'll just add two things. One is that, well, for birds, we're very unlucky in that birds do not preserve well, and many of their morphological features are quite conserved. So we don't have a lot of means to get ancient DNA, relative to mammals. But one of the fun things that I think has changed is there's some very useful and new statistical methods that allow you to directly place fossils in the phylogeny, combining morphological data and molecular data. And so that's a really powerful way, like Sushma says, to sort of try and better understand morphological innovation, but also to better date your phylogeny. And then the other thing I would say is that some of the new methods that are out there from the ancient DNA world have sort of enabled us to, not for ancient specimens, but for many specimens that are in museums around the world, you can very efficiently now obtain DNA from some of those museum skins. And in some cases, those specimens are over 100 years old. And that, for many birds, is 100 generations with a 1 year per generation. And so it gives you an opportunity to look at how genes have changed over time. And I think that'll be a future revolution that we're going to see unfold in the context of biogeography as well as other kinds of evolutionary studies.
Yeah, it's really interesting, Rauri, that you brought in the cultural evolution part with the bird song. Unfortunately, song doesn't preserve. So it's really fascinating to see how, how that can actually help, understand evolution. There's some other questions. Kadambari, I'm just scrolling down. Kadambari says that thanks to both Sushma and Rauri for taking us through this virtual travel across space and time through Africa. I think there are a few more questions for Sushma. There's one on the chronology of arrivals linked with paleoecology and climate, and if there's any kind of associations, I suppose, with frugivores, migratory birds, with habitat. Any links that you can?
Yeah, that's a great question. We haven't actually looked at diet yet. Most of the birds I've been working on are insectivores, and definitely want to look at how diet might be impacting some of these diversification patterns. Migration is kind of interesting in Madagascar. It actually is not a key part of the migratory route of most things. And so you have migratory pathways where things in the Palearctic go across Asia and get to Africa, and they skip right over Madagascar. So it isn't, there are birds that kind of generally move around from Africa into Madagascar, kind of widespread raptors and water birds, but it isn't like a place where a lot of migrants go. So I think it's kind of another illustration of how isolated Madagascar it is. You know, like in many islands, species accidentally get there, and once they get there, they can do things they couldn't have done otherwise. But it's most of island biogeography, I think, is starting to be seen as more catastrophic events rather than something that we can model easily. And that addresses this other question that
there's
Vivek
some
had.
questions on. Sorry,
I was just going to say there was one other question about the island biogeography models, and I don't want to be too disparaging on any of those any of that research, but you know, a lot of the kinds of predictions we can make from MacArthur and Wilson it doesn't work for a lot of islands, and definitely doesn't work for Madagascar in particular. And part of that, I think we've talked about this in many ways, like they don't bring in speciation, they don't bring in evolutionary theory into their models. And so that's primarily the reason why now when we're talking about biogeography, we should be talking about evolution and ecology and the interplay of that.
Yeah, Sushma, could you also just maybe mention something about what species are closely related between Madagascar and India? Because I see this question on hemipus, if those are related between Madagascar and India. So that may be something that interests people.
Sure. I wish I had more time to like put in more of these beautiful bird pictures somewhere. So yeah, hemipus is actually a close relative of vangas, and when we kind of had to make this call when we were publishing our paper, where traditionally the term Vangidae, the family name, was for the Malagasy lineage, but then when we worked out the phylogeny, there were these 6 kind of little known species, genera that were from Asia and Africa that were closely related to vangas, and so we kind of brought them into the family, and so now hemipus is part of the family of Vangidae, but the Malagasy vangas are the kind of original adaptive radiation that we talked about. So hemipus, the wood shrikes, let's see, there's a Hypsipedes, the black bulbul, that is also closely related, that's in the Western Ghats and other parts of India, that's also closely related to the species in Madagascar. And that's something that we again need to spend a little bit more time looking at phylogeographic structure, because it seems like there's probably more lineages there, more distinct lineages there. The forest owlet is also closely related to well, that's also a taxonomic mess of things that the species on Madagascar was placed into like all these different genera, and now it turns out it's closely related to the forest owlet in India. So that's another example. I'm racking my brain. Can you think of anything else, Robin, that I haven't mentioned?
No, this is, that's really good list. There's one question. Yeah, sure.
With a quick question for Rauri, because it's not letting me type in my question, so I'm going to have to ask it to you. So in the Eastern Arc region, you get a lot of monsoon winds, right? So it's a bi-monsoonal pattern.
Yes, it is. Yeah.
But aren't those winds strong enough to carry a lot of things across those distances? Even that large one, okay, maybe not the largest 100-kilometer one, but I'd have thought there'd be a lot of repeated movement with the monsoon winds.
The thing that's fascinating is that whole mountain chain is only about 500 kilometers, right?
Yeah.
It's tiny. And some of those forests are the size of a university campus. So they're very small.
Okay.
but they're very old. And so I think that you're right that the monsoonal winds are incredibly important because they actually what keep the forest there. They're so important. They bring the rain and maintain the forest. But I think what's happening is when you look at the ecologies for the different birds and how structured they are. So if we compare an understory bird, we see these very, very deep levels of genetic structure. But I've also done some examples where we've looked at birds that feed in the canopy, and they less geographically structured than some of the understory. So it's possible that, you know, those birds may be more prone to getting windswept or something. But even so, it seems to be a remarkably rare event.
Yeah.
Despite the monsoonal winds. Yep.
Yeah. Okay.
There's
Thanks.
another question. Yeah. Sorry. So there's another question which I'd like to kind of interpret slightly differently. So this is about Sky Islands and Madagascar being these isolated biogeographical locations. So how does multi-species interactions, you know, at the community level, how can that affect some patterns? And I suppose, Rauri, if you could elaborate on that as well.
I'll have a go. I think the honest answer is I don't completely know, but it's something that we're starting to try and work on, especially now that we can. I guess there's two insights I can give you. One is I'm fascinated by these old species and young species in some of these mountains that make up the communities. And we can reconstruct, because we have such good phylogenies now for many birds, the ancestral ecologies. And in many cases for these old species, we see that they've actually shifted ecologies over time. They don't maintain, even though they're very old, they've shifted their ecological niche. And then the other thing I can say is in some communities where we get multiple species, so for example, in sunbirds, the variance in traits like bull length or bull size gets very small as you add members to the community. So it's like each one specializes more. And if you remove a member from the community, you see a rapid change in traits. So as a simple example, with these double-collared sunbirds, they can vary from a bird that's 6 grams to a bird that's 12 grams. If you remove the large one, the small one will often increase in size up to 8 or 9 grams if it's on its own. So you see these release or constriction of traits. So that's something that I'm trying to explore a lot more now, but yeah.
Wow. I have another small question, which is on the song and the age of lineages. I wonder if you would find some differences in patterns depending on one of those new divergences versus these old divergences where you have a lot of time for changes to happen back and forth. Do you have any thoughts on that?
Yeah, I don't know. I certainly think that jumping into a new, I mean, intuitively, I would think if you colonize a new area, you're likely to change in song pretty quickly. But I'm no longer convinced that that's actually what's happening in these very old mountains. And I'm thinking of plumage as well, because it's the same, where it seems that some of these birds have been there, 3 or 4 million years old, and yet I cannot distinguish them. I cannot tell them apart from another population on another mountain in any trait. And so I have to think that stabilizing selection is actually been vastly underestimated in some of these. And I kind of think about it as much of the work in the past has been focused on North America or Europe. And there you had glacial ice sheets that kind of, I think of it as wiped the slate clean and things started again. But in some of these tropical environments, history layers on top of itself and it never gets eroded. And that's true for much of the tropics. And we only now just beginning to, I think, start to reveal that. So I don't know, but I think it's a really interesting area to work in. Yeah,
this has a lot of parallels for the kind of work that we're doing in the Western Ghats as well.
I mean, it's similar to what you found, with some of your Western Ghat birds in song as well. So it's not uncommon. Right. We just haven't paid attention to it as much.
That's right.
Okay.
Right. Thank you.
Thanks. Yeah. Thanks, everyone. Thanks Sushma and Rauri. Great talk.
Thank you.
Thanks, Sushma. Thanks, Rauri.
Thanks, everyone.
Thanks,
okay. Bye-bye.
Bye.
Yeah.
Bye, everyone.
Bye.
Look forward to seeing everybody tomorrow. Yeah, we start half an hour earlier tomorrow, so please note the change in time. It's half hour earlier than today's start.
Okay. Bye. Bye.
Bye.
Bye.
Thanks,
Bye.
everyone.
Thank you.
Bye.
Thanks, everyone.
Bye, Vivek.
Bye-bye.
Bye.